The elephant. Graceful, elegant, massive, and cancer-resistant. But how? In humans and in all animals, cancer occurs when DNA is damaged by radiation, pollutants, or is miscopied during mitosis, causing cells to divide at a rapid pace, causing the growth of tumors. The larger the animal and the longer an organism lives, the more likely that the animal is to get cancer. Which brings us back to elephants, which despite their massive size seem to get cancer at much lower rates. How is this possible? It's all in their biology.
Cell communication is essential for all aspects of life, including the prevention of cancer. Cell communication falls into 4 categories: paracrine signaling, which is when signals travel short distances; autocrine signaling, when cell signals travel to themselves; endocrine signaling, when cell signals travel long distances and through signaling with direct contact. Basic cell signaling includes the release of a ligand through exocytosis, which is active transport using ATP, which then binds to a receptor protein on the target cell membrane. The cell membrane is comprised of a bilayer made up of lipid macromolecules, which are hydrophobic or don't have a charge, bonded to a phosphate, which is hydrophilic or is charged. This creates a selectively semipermeable membrane, which allows in nonpolar molecules such as O2, N2, and CO2 through the phospholipid bilayer through the process of diffusion, or larger or polar molecules through transport proteins through facilitated diffusion using ATP, which is made using oxygen and carbohydrates through cellular respiration. Phosphates cleave from ATP through hydrolysis or the breaking of hydrogen bonds with a water molecule attached to a protein receptor, which when activated causes the phosphorylation of chemical messengers, which relay the message to induce a response, such as cell growth or even cell death.
Proteins coded by DNA, which is comprised of a sugar phosphate and nitrogenous base of either adenine, guanine, cytosine, or thymine, can be located on the membrane, or there can be others located around the cytoplasm to be used for various processes when needed. Proteins are made through protein folding in ribosomes, in which there are 4 different stages that are essential to a protein being properly created. The primary stage consists of the aforementioned amino acid chains. In the second stage, pleated sheets or helixes are created through hydrogen bonds that fold the amino acid backbone. And in tertiary interactions, R groups and side chains that may differ in their polarities and repel or attract each other may create the structure through certain bonds, such as disulfide or hydrogen bonds. The quaternary stage consists of multiple amino acid or polypeptide chains. The intricacy of protein folding allows for certain mistakes that may greatly affect a protein's ability to function in the cell. Denaturation of certain proteins after exposed to a change in its environment can sometimes be fixed through refolding the protein, however. Before it is fixed, or when the change is permanent, the protein becomes unusable. Proteins, more specifically enzymes, can be denatured in this way, and it affects their ability to lower reaction energy and speed up cellular processes. Getting back to cancer in elephants, in most animals, the TP53 gene codes for the p53 protein that binds onto DNA to repair damage among other functions and acts as a tumor suppressor. However, typically most animals only have 1 gene to code for this essential protein. If this gene is damaged, the p53 protein will not fold in the same way, and the misshapen protein will not be able to bind to and repair DNA. Elephants, in contrast to most animals, have 20 copies of this gene and are able to not only create more of the p53 protein, but also can account for any damage to the TP53 gene by having backup copies. In addition to the large quantity of TP53 genes, elephants also have the resurrected zombie gene, LIF6, controlled by the p53 protein, which can induce apoptosis or cell death when the cell is damaged beyond repair.
In a normal cell, the p53 protein that is floating around in the cytoplasm is deactivated by MDM2. In elephants, when the cell DNA is damaged, sensors send a signal ligand to 2 kinases, ATM and ATR, which then phosphorylates the p53 proteins by binding a phosphate to serine 15. This allows the p53 to activate and bind to the transcriptional activator CBP/P300, which allows for increased transcription of genes into proteins. Downstream, the P53 protein also is phosphorylated through the binding of a phosphate through dehydration synthesis and onto the serine 20 by two serine 309 kinases, KHK1 and KHK2, also activated by DNA damage. The activation of P53 in this case protects the P53 from being inhibited by MDM2 and allows it to accumulate in the damaged cell. The protein is then able to bind to DNA to activate the gene that creates the protein C21 to arrest the cell cycle in the S phase or interphase. This allows for the damaged DNA to be repaired by other proteins. If the damage is severe, the cell will trigger apoptosis or cellular suicide. Although the use of P53 is common to most animals, elephants in particular also have the LIF6 gene, which when activated by the phosphorylated P53 protein creates a protein which